We're very fortunate to have Dr. Peter Salamone joining us today. Peter is the Technical Manager North America for Laffort USA. He has broad experience in industrial biotechnology in the wine industry, both in the production setting with Ian J. Gallo Winery and Diego Chateau and Estates Wines, as well as wine industry service experience with enological product and enzyme vendors. Coupled with his diverse production winery exposure, Peter's depth of biotech experience in molecular biosciences, including microbiology, enzymology, and genetics, provides Laffort USA with an outstanding technical resource. So today, Peter is here to join in with us to talk a little bit about Sur Lie wine production. So with that, Peter, please join us. Thanks.
I'd like to thank the folks here at CCOVI for having me up to talk with you guys today. Got a bit of an interesting topic, and what I'm going to talk about is Sur Lie in kind of a general way at first, and then some specifics. I mentioned up here the word science and wine character. This isn't a lesson in winemaking. It's a lesson in the investigation of what winemaking practices are really going on molecularly. It's what we pride ourselves a lot on at Laffort. We do a lot of front-end R&D. We publish all of our findings. We share it with the industry, and it drives what we do as a service component in helping people understand what they're doing better, so you have more control over your processes and then can use things to give advantage to your wine. So, real quick review: Sur Lie, aging, batonnage. Going back a few years to the Roman days, Roman historian Cato credited in historical documents with realizing that wines that were left on their lees developed different flavors than those that are rack clean. Given the 6,000 years humans have been making wine, that was 4,000 or so years into the process, so he was a real visionary. Forward a few more years when practices in France advanced, and the terminology for what we're talking about today, Sur Lie, is a French term for leaving the wine in contact with its lees, and also batonnage, which goes along with Sur Lie, aging, is the term for stirring the lees back up into the wine. So the classical Burgundian schedule for Sur Lie cellar aging, racking off of the gross lees around November, December at the end of primary fermentation, racking again in March, end of winter kind of thing. And then rack again about 3 months later, June, weather getting warmer, more chances of microbial issues, so a little CO2 in there, or sorry, SO2. And then about a year later in September, rack it off again and consider whether you do further cellar aging, bottling. So I think everyone is fairly familiar with the process of Sur Lie and what it really means, but there's a little bit of historical background. And why? Why would people do all this? There's got to be some tangible benefits, and over the years, lots of benefits have been noticed empirically, enhancing the structure and mouthfeel of wines, extra body and decreased astringency, increased aromatic complexity, flavor, aroma, depth, and length. I wouldn't say that all of these things are characteristics of every wine that's ever been Sur Lie aged, but throughout the different varietals and practices, pretty much all of these things have been documented in literature. Increased perception of sweetness, the stability of color, proteins, tartrates, protection from oxidation. More recently, looking into the nutritional aspects of malolactic fermentation and Sur Lie's benefits in that respect, and improved finding and clarity. But as you can see from these nice display barrels, when you stir the lees, you get a lot of stuff, not all of it, just yeast cells that settle, but also some other particulates that can be in the wine. And so it mixes everything back up again. So there may actually be some risks involved. Otherwise, with all these benefits, why would you ever make wine any other way? But there's a chance with those solids in the bottom of the aging vessel, whether it's a barrel or whether it's a tank, that you can get some reductive aromas, some hydrogen sulfide or some mercaptans. And when yeast get embedded down in those lees and they get unhappy where they are metabolically, they tend to do things that let you know they're unhappy. Oxidation from the frequent stirring, long storage in vessels that have the ability for oxygen to ingress. Microbial sanitation, again, long time storage with solid particulates. There's always the chance that a guy you don't want growing in there is going to grow. And like I mentioned earlier, the improved nutrition for MLF, it's kind of a double-edged sword. Saccharomyces can throw off some medium chain fatty acids that Oenococcus does not like. So there's the possibility too that you could develop inhibition of your malolactic. So taking it a little step closer into what's going on in these vessels and in the bottom of these yeast, what's really happening is a process we call yeast autolysis. And that occurs at the end of the alcoholic fermentation. Everybody's usually pretty familiar with fermentation curves: lag phase, log phase, stationary phase, death phase. And then, so at the time when the cells start dying off, they kind of lose their vigor and metabolic activity, and so they'll tend to settle down in the tank. Even some live yeast will as well. And so at the bottom of the tank, they're subjected to physical pressure. Larger tanks, or even in barrels, hydrolytic enzymes and oxidative damage can degrade the cell integrity, and you release the components of the cells into the matrix, which is the wine. And so, wrong button. So what ends up coming out of these guys? Peptides, fatty acids, amino acids, nucleotides, polysaccharides. That sounds like a list of pretty much everything that's in there in a living cell, but what does that really have to do with wine and the effects of Sur Lie aging? Certain of these categories, there's been some definitions as far as what these contribute. You can see flavor, aroma, sweetness, nutrients, antioxidation. We're going to talk about peptides and their specific contribution to sweetness and antioxidation today. Amino acids, again, conjugated to other molecules, create flavor, aroma molecules, provision of nutrients for the Oenococcus, for the MLF. Nucleotides, again, there's incidents in the literature where people describe certain nucleotide derivatives as sweet or having flavors. And again, nutrition for MLF. Polysaccharides, much of the stability things that we spoke about earlier were provided by different polysaccharides. That's one of the focuses of talk today. We'll get into that in detail, and mouthfeel. And then just, I left in the middle antioxidation and fining. That's kind of a property of the yeast cell wall membrane fragments. I don't want to call it just yeast hulls. That's kind of just the cell wall. But through this process of autolysis, you can get cell wall fragments that still have membrane attached, and that contributes a lot. We'll talk a little bit about that at the tail end of this with specific adsorption properties of those molecules. So, looking a little closer, here's a diagram of yeast. I think everybody is familiar with our friend Saccharomyces and the general unicellular eukaryotes and what kind of organelle structures they have inside them and all. The classes of molecules that we just spoke about: cell wall nanoproteins, cell membrane associated peptides, cytosolic peptides, and sulfur-containing amino acids, cell wall membrane fragments. They all come from in here. But what I wanted to talk about a little with this diagram is to do a little cell physiology kind of explanation about things like the endoplasmic reticulum and the Golgi apparatus and peroxisomes and vacuoles and mitochondria and all these intracellular compartments that enable cells to carry on the metabolism that they do. Without that segregation, the biochemical activities can't occur the way that they do occur. And what happens when nutrients are depleted and the yeast kind of give up, there's so many of them and they go into death phase, that you start to lose integrity inside here first. So you get some release of contents of some of these normally discrete areas, and you can get hydrolytic enzymes that are functional in one area, get exposed to others, and you start to get degradation on the inside. And then when the yeast stop their metabolic activity, everything that has order, like a cell, takes energy to keep the order. When you stop processing the sugars and you stop making a lot of ATP, cellular integrity can give up. And what happens is at the cell membrane and cell wall, you can start to get some degradation too. So, a little closer picture. So you've got your cell membrane here, and you've got your cell wall out here, and you've got some polymeric hydrocarbon, basically chains that kind of hold everything together out in the cell wall, and you've got your nice protein mosaic model of your lipid bilayer here. There's proteins associated at the inner surface. There's proteins embedded here. There's all these lipids. They all take maintenance to be maintained. And there's a lot of nanoproteins in the outer cell wall. These are all drawn very uniformly, but all these carbohydrate chains that are exposed on the surface of yeast, that's kind of how they communicate with other cells and with their environment. And these chains are actually quite diverse. And we'll see some of that as we move forward in the talk today. So I've got this talk broken up into a few different parts. That's just a quick introduction. I wanted to talk a bit about yeast, talk a little bit about overall the cellular components as we move into what these things do and specific identification of the molecules that contribute to the Sur Lie characters that we see when we do that form of winemaking. And so I just wanted to, I'm going to be presenting the works from a published paper, from a patent, and then just from some other research that's been done at ISVV and at Sarco Lab in Bordeaux. And so I just, instead of putting all the names at the end, I wanted to put them here before I start my first part. So I'm not going to go through everybody's names, but just say that at Laffort, we really value research, innovation, and quality. It's something we talk about all the time, and it's what drives our front-end business. That's why I like to work there. I'm a scientist at heart. So today we're going to talk about three main areas of Sur Lie and the work that we've done on defining this. We're going to go into some peptides in wine and show a paper on the characterization of that. Specific nanoprotein characteristics, and that's pretty much the information that's been distilled down and is published in a patent. So I'll be working off of that information. And then some information on antioxidation and fining. So peptides in wine. This work was published in the Journal of Agriculture and Food Chemistry, as you can see, in 2011. So a fairly recent publication. And the aim of this work was to validate the role of yeast leaves on the increase of sweetness. It's an observation. If you do Sur Lie aging, the wines can take on a sweet character. People have observed it, nobody's defined it. So we wanted to take a look at that and validate it. And then we're looking to identify the chemical or biochemical origin of that. So perception of sweetness in leaves on a really broad validation. What we wanted to do is to prove that it is a property of the leaves. So we started with a base wine, 12% alcohol, 6.9 grams per liter glycerol, a little bit of glucose fructose left in the wine, 0.37. So that's your base wine. The lees in this study, you can see we're looking at ethanol, we're looking at glycerol, and we're looking at lees. So let me just define how we made the lees. What we did is we grew the yeast, we harvested the yeast, we washed the cells, and resuspended them in a model wine solution, and then held it at 32 degrees for 10 days in the dark to allow autolysis to occur from these lees. So this is a bit of an eye chart, and I'll walk through this kind of slow. These are actually two tables, but you can see that for this particular sensory test, what we were doing is a forced ranking. We prepared 4 different samples for these different variables, and we asked panelists, 38 panelists, to rank them 1 through 4, sweetest to not sweet. And then we added the 1 through 4 numbers up for each of the 38 panelists for each of the different modes, and we took a look at the totals. So let me just go through with the ethanol. What we did with our base wine, we added 0.5%, 1%, and 1.5%. So we had the 12% base, and then we had these additions. And you can see when we did those and had everybody taste those and look back at what the rankings were using this wonderful statistical formula that I'll talk about for 30 minutes. No, just kidding. It was not a significant difference between the 4 modes. So the alcohol in those concentrations didn't contribute any sweetness as detectable, statistically significant. So the same with the glycerol, same number of panelists and all. We took the base wine, we added 1 gram, 3 grams, and 5 grams. Did the tasting, ranked them the same way, did the statistics, no significant difference between the 4. So up to 5 additional grams of glycerol did not add any detectable sweetness to the base wine. Then we took our lees, and I'm sorry, I described the wrong way that we did that. I have to backtrack on that. We took the red wine base, we grew the yeast cells like I said, we washed them, resuspended them, counted them, okay, and then added back this number of cells to the red wine and carried it through the 10-day autolysis procedure that I mentioned. So these wines with 2 times 10 to the 8th, 4 times 10 to the 8th, and 8 times 10 to the 8th cells per mil that had gone through the autolysis process, the 32 degrees, 10 days process that I mentioned. We had them tasted by the panel as well, and with these numbers, 1 through 4, there was a statistically significant difference. It was detectable that the higher cell count wines did have a perception of sweetness. So that's a validation that lees is what's contributing to this sweetness. It's not differences in alcohol or differences in glycerol. When you Sur Lie age wines, there's a component in the Sur Lie character from the yeast that lends sweetness. So that's the first step, just to validate that it is indeed from the Sur Lie practice. So what we wanted to do now is to take a look at what component could that be. And so in order to do that, instead of taking a wine, which has got, everyone knows the chemical complexity in wines and how difficult it is to analyze specific compounds in it. What we wanted to do is take a model wine solution that's less complex, grow the yeast in it, grow the Saccharomyces in the defined media, the model solution, harvest the cells, wash, resuspend, do the autolysis, and then check the autolysate. And what we did is we started with an ultrafiltration regimen to separate that into fractions. So here's the process that we used. You can see that we took the fermented model medium with the autolysis, and then we did an ultrafiltration through a 10 kD filter. We had a 10, then every molecule bigger than 10 kD was retained. We subjected the filtrate to a 3 kD filter, so the retentate was between 10 and 3 kilodaltons. Then we subjected this filtrate to a 0.5 kilodalton nanofilter, so the retentate here was between 3 and 0.5 kilodaltons. And then we had the final filtrate, which was molecules less than half a kilodalton. So what that left us with is with 5 different fractions to take a look at and see which of these fractions contains the sweetness component, if it is isolatable. So here's where we did things a little bit differently. Instead of a forced ranking kind of test, we did triangle testing to see if respondents could pick out the one that was different. And so what we found, and of course we were asking them for, which one is sweeter. So they would, in this particular thing, you can see that the yeast lysate autolysis medium, the crude extract, that there was statistically significant identification of the sweetness in the lysate. And then in the 10 kD fraction, it was not significant in selection. 3 to 10, not significant. The 0.5 to 3, significant. So the molecule we're looking for is this, and again, in the smaller fraction, it was not significant. So the property isn't shared between these different fractions. It's in the 0.5 to 3 kilodalton fraction. So that's pretty interesting. I mean, we've got it down now. We've cleaned it up a little bit. We've got a certain size, but we still don't really know what it is. Could it be, could be nucleotides, but that's pretty big for nucleotides. But still, so we asked, the premise was that this could be a peptide. So we asked ourselves, how could we show if it was a peptide or not? So what we came up with was to do an enzymatic treatment of the 0.3 to 0.5 fraction and see if the treatment of that fraction resulted in any differences in the sensory perception. So we did the protein. What we did is we made a concentrated solution of that 0.3 to 0.5, and we did the sensory evaluation, a triangle test, again with 23 respondents, and we asked between the synthetic solution and the solution treated with the proteinase K, was there a difference? And there was no significant difference between the two. The sweetness had gone away from the fraction that was previously identified. So with proteolytic treatment, the sweetness went away, told us that what we're dealing with is a protein or peptide of somewhere between 3 kilodaltons and 0.5. So next step, we went to HPLC separation. We did a HPLC HR column. We took this 34-minute peak, the majority peak, and we then separated it on a reverse phase C18 column. And then we took, again, the most populous peak, and we did some online capillary HPLC nanospray ion trap. Basically, we sequenced these peptides. And so we took the sequence of the peptides and blasted them to see if any of them were identifiable and where they came from. And there was a number of different yeast proteins that were represented by these peptides, but most of them were part of the HSP 12 kilodalton heat shock protein. This is an inner membrane associated heat shock protein. It's like a chaperonin. It helps keep proteins in their native conformation. So when this protein is not just responsive to heat, but ethanol stress will also upregulate it. And it works on I don't know the exact targets, but pretty much it works on the inside part of the membrane to keep proteins in their conformation and keep the membrane integrity intact when the cells are challenged environmentally. So there's a bunch of that in there, and so we had this idea. Now we've got an idea that it might be the HSP12. It's the majority, but it doesn't mean that that's it. You can see there were a number of other peptides that were identified as well. And so we had a choice here. Did we want to try to do more biochemistry and try to purify some of these things and test them for sweetness? Or is there a way we could get at this a little more efficiently? So what we came up with is a genetic strategy. What we wanted to do is, the hypothesis is that the HSP12 peptide is the source of the sweetness. So what we were going to do is develop a genetic knockout in Saccharomyces and see if that didn't remove the sweetness character. So here's where it gets fun. So the genetic approach is kind of interesting. It's a lot of molecular biology and a lot of yeast genetics, but it's pretty elegant and it's very well done. They did a really good job with this. So the Saccharomyces strain FX10 is homophthalic, fully homozygous diploid strain. The X strains from Laffort are all bred strains. We use micromanipulators and we've created these strains through native breeding, but selective breeding. And so we know the genetics on this guy really well. So we started out with that. And just through usual yeast manipulations, you can create a haploid strain. Now, diploid, of course, is 2 copies of all the chromosome, 2 copies of every gene, and haploid is a single copy. So we wanted to create a haploid, which yeast can do, so that we could genetically manipulate that without having to worry about having our manipulations be on both genes. So first thing was to create the haploid, then we used the Redox recombination system to create a knockout in HSP12. There's a bunch of steps in here that are in the paper. I'm not going to go through all those today, but if you're interested in molecular biology, it's in the paper. So we knocked out HSP12 in a haploid strain. Then we crossed the HSP12 with FX10 by micromanipulation. And then we segregated those and allowed self-diploid formation, which is a property of having the HO endonuclease and the mating factor genetics of Saccharomyces. And then we verified that we had a homozygous delta HSP12 sporulation on selective media as well as PCR. So we created a homozygous knockout for HSP12 in the FX10 genetic background. So what we wanted to do with that, now that we generated that, was to, let me go through. This is just a list of the strains FX10 and the different constructs as we went through the haploid, then the knockout all the way down to the homozygous deletion for HSP12. So what we wanted to do with this is detect, if we developed one of the, if we did the autolysate with the FX10 and then did the autolysate with the double knockout, then we wanted to do a sensory test. Again, it's a triangle test to see could the respondents detect a difference in sweetness between the FX10 and the delta HSP. And again, you can see statistically significant, yes, the FX10 that was not knocked out was noticeably sweeter than the one that was knocked out. So from all the very beginning of sweetness in lees to determining exactly what the factor is, which peptide, what protein it comes from, pretty elegant work, and it definitely shows that it's not ethanol, glycerol, additional sugars. So I've got this talk broken down into basically 3 sections. And that's, well, I'll go through the summary, and then if anybody wants to discuss any aspects of this, we can stop at this point and go through this before I move into the next one. But just as a summary, again, validation of the sweet effect of the lees, determination of what molecule it is, showing it was a protein, purification, identification, and then again confirmation that we had the right target.
Other than proteinase K digestion that followed through to sensory tests, or did you just focus on the proteinase K because it gave good results?
Well, we used the proteinase K on the hypothesis that the sweetness was caused by a peptide. Right. So we weren't really looking for a sensory impact from proteinase K other than the absence of the sweetness.
But I just mean, did you do? Because you thought it might be a peptide. Did you do any other tests thinking it might be something else?
I didn't design those experiments. I can't say what they did that didn't make it into the paper.
Okay.
Sorry. Well, even though if they would have done 37 other tests, the fact that the proteinase K broke it down, defines the target. And the fact that when you, especially with the deletion mutant, when you take that gene away, there is no sweetness. So it's not a secondary metabolite. It's not a different compound. I think it's pretty conclusive that it actually is the peptide.
I was just curious if there are any failed tests.
Yeah, there always is. That's why I call it research. Yeah. Now, I think that people had thought for a long time. There's previous publications of peptides and their sensory impacts. but there's been no publications of peptides in wine and sweetness. This was the first publication that identifies a peptide in wine as having sweetness. There's lots of publications, flavor chemistry journals about peptides and their impact. So when we started looking at things like the sweetness, and you realize that it's not ethanol and it's not glycerol, when you look at the things that people associate with sweetness in wines and in the wine matrix, it just kind of pops up as the next most likely target.
Peter, are you thinking that these peptides, these proteins, do they possibly have carbohydrate attached to them, or are you thinking it's just the peptide unit itself that's eliciting this sweetness response?
Yeah, it's just the peptide. From the nanospray, MSMS, if that had carbohydrate attached, we wouldn't have been able to get sequence on it. They have to be just peptides at that point. And then with the subsequent work showing the HSP predominance, and then the genetics showing that it is from HSP, which is an inner surface protein, it's highly unlikely that that's glycosylated. Cool. Well, let's see what else we can find out. So that was peptides. We're going to talk a little bit about manoproteins. And everybody's heard, Sur Lie aging, and probably the most predominant thing people say is the mouthfeel. You get great mouthfeel, and it's creamy and rich, and people say, oh, it's manoproteins. It's manoproteins that give it the mouthfeel. And that's pretty much the way the wine industry works for hundreds of years, if not longer, that observations are observed and ideas are put forward about why, and sometimes it gets investigated and sometimes it doesn't. So that's one of the observations. So we set about this to ask the question. I guess at this point I'll mention, we've had an initiative since the early to mid '90s at Laffort, well before I started with the company, about this whole concept of Sur Lie aging and the observations throughout centuries about the benefits that it brings. And so we asked ourselves, what's really going on? And it's a big question, and you can see just from the first paper that the elegant research that can come from it, including things like the verification of manoproteins. And you can see that out of Dr. Dubadru's lab, when Virginie was there back in 1997, they had published a paper concerning the role of yeast manoproteins in the stability of wines. And that was a very early paper. But as things moved along and people started looking at these molecules and preparations of the molecules, things were put forward for product use for manoproteins to be added to wines. And in 2005, OIV regulatory approval for addition of manoproteins. And it was a pretty big deal. In 2006 at Vinitec, there was, the Global Award for Innovation went to the manoprotein products for addition to wines. And then, it's advanced from 05 and 06, and later on it works its way into the popular press. So it gets disseminated out a lot of different places. But there's more to it. It's not as simple as manoproteins from yeast. What was going on initially was you could grow Saccharomyces, harvest the cells, do a heat treatment on the cells, and release a lot of the cell wall components that contain manoproteins, and that was what was used to create this effect that was really undefined, mouthfeel or stability, heat stability, protein stability, color stability, a lot of things attributed to these preps, and it seemed like it was a little too complex to be explained in a simple way. So some more work was done, and I threw this up here as the eye test. I'm going to have this sentence here. We'll have a test on at the end. This is a lot of the cumulative work that was done in specific identification and properties, much like the peptide paper that I presented. This was all disclosed in this particular patent. So, what we started out to do here is to take a look at the heat-treated yeast manoprotein fraction versus an enzyme-treated fraction, to take a look and see, are these things different? Do these treatments deliver fundamentally different preps from the outer surface of Saccharomyces when you treat them this way? This was the standard way of treating it to generate these preps. This was a bit of a different way that we worked out at Laffort. So in a general HPLC, you can see that there's a difference in the peak elution profile. This is just a void peak, and this is the eluent peak, and they're in different positions. So there's something fundamentally different there. That's really all we wanted to look at with that, is there a difference? So we took a look a different way. Because these things are polymeric carbohydrates, branched, they're, sometimes HPLC separations aren't the best way to get a general separation from those kinds of molecules. So we did a capillary electrophoresis. You can see again the heat treated versus the enzyme treated, and there's a real significant difference between those two. Again, this particular peak W. And fortunately, peak W was shown to exhibit protein and tartrate stabilization properties. So that's of interest. So what we wanted to take a look at is in some different wines using the heat-treated versus enzyme-treated fractions at the same amount. Does it deliver the property we're looking at? In this case, protein stability, as measured by a reduction in the amount of bentonite it takes to make the wines heat stable. So you can see that in reference wine 1, the heat-treated sample didn't have any kind of an impact, but the enzyme-treated in 3 separate fractions, basically 3 different enzymatic digests, it delivered the result, basically cutting the amount of bentonite needed in half. The same with another wine, 2 separate preparations of the enzyme-treated, cut the amount of bentonite by half. And in a third reference wine, again, 2 separate preparations, cut the amount basically in half. So the enzymatic treatment delivers a certain molecule that has this property. So a little more investigation to try to figure out exactly what it is. And I've cut this down because I don't want to belabor too much the purification process, this and that. But here's an ion exchange, resin DEAE, and here's an affinity chromatography, concanavalin A, which is a lectin. It specifically binds carbohydrates. So you can see that through the purification, you're increasing the percent of this MP32, 32 kD manoprotein. So as we move through this and characterize these fractions as having the activity or not having the activity, okay? This is the active fraction, and we're getting more and more of it as we go through these purification steps. That indicates we're on the right track with trying to isolate it. This is information from a electrophoretic SDS page gel, and you can see from the initial extract to the DEAE fraction that had activity, to the CON-A fraction that had activity in increasing amounts of activity. That from these 5 different bands that show up on the CON-A and all the others, this is the only one that increased in concentration. So the protein stability effect paralleled the increase in concentration of the 32 kD manoprotein, which leads us to say that the 32 kD manoprotein gives the protein stability effect. So, again, a lot of properties for manoproteins. Tartrate stabilization is another one. So, again, just a real quick experiment. White wine, rosé wine, red wine. Control, basically untreated wine that's unstable. The test was 6 days at -4 degrees. Does it throw tartrates or not? Hence the measurable being 3 asterisks. Not real quantitative, but it's qualitative, and you can see that the heat-treated fractions had little effect or no effect on tartrate stabilization, while the enzyme-treated inhibited tartrate stabilization at 25 grams per hectoliter. So again, we wanted to take a look at different wines and make sure it wasn't a one-off on the property. So 6 white wines, 2 rosés, 3 red wines. The reference wine, of course, was unstable, and then we used some metatartaric acid as kind of a positive control because it will inhibit tartrate precipitation. So we put that in, and I don't know why they didn't determine these, but you can see that it worked on these, and it didn't work on these red wines. The heat-treated did not work on any of the wines, and the enzyme-treated did. So we're seeing, again, a separation of the properties in the stabilization of tartrates, much like the protein stabilization. So I've named this up here already. So what we want to do is take a look at the enzyme treated, do a separation on HPLC. We want to take this, this is basically the void volume. We tested both P1 and P2, and you can see that the P1 had a very light effect, while the P2 gave a stabilization effect. So we were able, on this HPLC separation, to separate effective from not effective. And then again, we took a look through, again, I'm not going to belabor the purification. You can see we have P1, P2. I've got the Con A fraction over here again. The concentration increased in this particular fraction and not these other three. So again, with the carryover of the effect, the tartrate stabilization, and the increasing of this size manoprotein indicates that the 40 KD, 41.6, we just call it 40, MP40, the 40 KD protein, or manoprotein, is the one that has the tartrate stabilization effect. So the protein is a 32, the tartrate is a 40. And interestingly enough, when you test either of these manoproteins separately, they do not give a mouthfeel effect. So it's a completely different set of manoproteins from the cell surface in yeast, or something else that gives the mouthfeel effect. So one of the particular properties of using the manoproteins is that they're concentration dependent. It's kind of a colloidal activity. So if you don't put enough manoproteins in, then you can still have a lot of nucleated bitartrate crystals, and you can get crystal formation that becomes visible and it's not stable. Or you can get the stoichiometry right, basing the dosage on the instability of the wine, and then it's balanced and it works just great and it's inhibited and it's basically irreversible and permanent. Or because, like I mentioned, it's kind of a colloidal stabilization, lots of molecules accumulate in soluble associations. If you add too much of the manoprotein, if you overdose it, if you just say, oh, a little is good, let's put a lot in, it'll tend to aggregate with other fellows in itself, taking itself out of concentration, and then you don't have enough to stabilize the wine. So if you overdose it, it could not work. So I'm just going to talk real quick about the mechanism of crystallization. Of course, with bitartrate, there's a balance between forms of tartaric acid, whether it has potassium on one end and a hydrogen on the other, or two hydrogens on both. Tartaric acid is a dicarboxylic acid, so you've got counter ions at each carboxyl group. But the only one that forms crystals is the bitartrate. It has hydrogen as a counter ion on one end, and it's got potassium on the other. The other guys don't crystallize, but the bitartrate does. And so the crystals grow in a characteristic shape. It's an orthorhombic, and you can see that it's nice and has specific faces, the 0, 1, 0, big flat face. But so what happens when you add the manoprotein, the MP40 in, these crystals nucleate, but the manoprotein attaches itself to the surface and it doesn't grow. These faces don't become defined, the orthorhombic shape doesn't take place, and the crystals stay very small and they never precipitate, and you don't see them in the wine. So with the MP40 and the tartrate stability, the first natural treatment to stabilize wine. Naturally present in wine, it's MP40. It's a native molecule in Saccharomyces cerevisiae. And again, the inhibition of the crystallization, which is very different from previous ways to stabilize wines, which were always subtractive. You took away the potassium ions, or you took away the tartaric acid, whether it's electrodialysis or whether it was chilling or seeding with bitartrate and further chilling. You're always taking something away from the wine. So using this kind of a strategy, I always thought this was something that winemakers would really love, because you make your wine, you nurture it through, you spend a lot of time in the vineyard and you taste your fruit and you get it just right in a number of different ways. You come in, you make your wine, and then when you go to stabilize it, you drop all that acid out, you drop the potassium out, you change the balance, change the pH, and you've got to come back and rebalance your wine. So I always thought winemakers would really love the fact that they could do this. They could make their wine and then stabilize it and not have to worry about rebalancing it again. Again, pretty straightforward. There's lots of advantages. Environmental benefits. This is where using this as opposed to traditional cold stabilization in the cellar, it's pretty rapid, so you save a lot of time. You're not throwing all your tartrates, so you're not having to clean everything off your tanks. It saves a lot of water. In larger facilities, that's very significant. And there's a big cost benefit in terms of energy in the wineries as well. So it's a really green practice to move away from the traditional cold stabilization. So, as an offshoot of this, I've just got 3 quick slides. Now, I mentioned the balance between the different forms of tartaric acid. And the tartrate stabilization, traditional cold stabilization, or membrane-based electrodialysis as subtractive techniques. And then non-subtractive, the MP40. And this is what I wanted to talk about for a second, just because the discovery of MP40 in the nanoprotein and the branch carbohydrate molecule that it is, and the fact that it inhibits tartrate crystallization, made people start to think about what other kind of polymeric molecules could have an effect like this. And we're seeing what's happening with the nanoprotein. And some work was done on a bunch of different polymers, and CMC, carboxymethylcellulose, was identified. It's a molecule that's been around for decades. It's in the food industry. It's a thickener in toothpaste and ice cream and salad dressings, and so it's pretty ubiquitous. It's a real, it's a bulk commodity. Hercules makes this stuff by the train carload. But in testing, this particular molecule, kind of an eye chart here, but it's pretty simple. It's carboxymethylcellulose. So you've got a cellulose backbone with your beta-1,4 linkages between the sugar units. You've got a carboxymethyl group that's just, here's your methyl spacer and here's your carboxyl group. And you got some sodium counterions in here. And I put a pKa of 4. This particular carboxymethyl group on this chain has a pKa of 4. What's important about that is, well, what's the pH of your wines when you're making wine? Above or below 4? Hopefully below. Yeah. So at wine pH, this molecule is going to be dissociated, and it's going to be negatively charged, which is important. I won't go into a lot of all this other stuff, but really, so how does CMC work? I mean, we don't know mechanistically how the nanoprotein works. We haven't done electron micrographs. We haven't seen the interaction. Haven't done things like microcalorimetry or using Biocore specific interactions to try to look molecularly what's happening at the crystal surfaces. So, but what we can determine with a molecule like this that we know a little bit more, like with the carboxymethyl groups and those negative charges all lined up along that linear polymer of cellulose, when the bitartrates start to lay down in a crystalline form, remember you've got a hydrogen at one end and a potassium at the other. Size-wise, the potassium is a lot bigger than a hydrogen. On the surface of this, these guys stick up like little speed bumps. Okay? Positive charges just sticking up all over the surface. And we've got those nice little negative charges all along the chain length of the cellulose fiber. Those things sit down on here like Velcro. So why it's not a covalent interaction, it's just an electrostatic interaction, but there's so many of them that the on-off rates for the individual interactions, they never come off. They get on there, and individually some of them can dissociate, but the molecule itself doesn't come off. We've got wines that were treated with CMC we've had for over 8 years now, and they're still stable. It's effectively permanent. The molecule is very stable in wine, like the nanoprotein, same thing, long-term, effectively permanent. So, that was kind of an offshoot of the nanoprotein work to find another polymer that acts that way. Nanoproteins. Comments, questions, discussion? Yes?
Okay, so you have something you can add, the nanostat, right? For the stability.
Sure.
Does it have the 32 kilodalton nanoprotein in it as well?
Yes. But you're talking about a specific product. The product is formulated to be added at a certain weight per volume based on the instability of the wine to give tartaric stabilization. Now, what benefit you receive from the MP32 and the protein stabilization, I think you'd have to quantitate that. And as you saw with the MP32, it doesn't give complete protein stabilization. It just reduces the amount of bentonite you need. So each wine, whether you treated it with the Manostab MP40, you'd still have to run a bentonite test and see how much is it going to take to get you to complete protein stabilization. It should be lower than a wine that's not treated with it, but we don't quantify the MP32 amount in that product. It's an MP40 quantitated.
Pete, yes. Question. In the Sur Lie aging, where some of these nanoproteins would also be released and interacting, do you also find some of these same results that are coming from the yeast as opposed to an additive to the wines, that you get better protein stability and better tartrate stability without having to go extra steps.
Absolutely, yeah. That's, basically the initial observation was that wines that were left on Sur Lie for a long time showed these characteristics. They never gave complete tartrate stabilization, but it would reduce it, and they never did complete protein, but it would reduce it. So yeah, these benefits are available just from Sur Lie aging with the yeast that you do your ferment on.
But from this work, you've now also been able to isolate the actual fractions from the yeast, and there are products now that are just based on these components.
Right, yeah. You can, basically what we've been able to do is show dosage-wise that you can get a complete tartrate inhibition. And so from processing the Saccharomyces and, basic QA work in product development, you pull enough of the active molecule into your finished product to dose it appropriately. And so yeah, like I said, it's the good thing about it is it comes from Saccharomyces. It's in everybody's winemaking today. Well, not the CMC, but all the manoproteins are, and just by Sur Lie aging your wines, you can start to gain these benefits, as well as, like I mentioned, the mouthfeel. These specific components don't bring mouthfeel with them. You have to put them in at really, really high levels, way above anything recommended to even start. And then I'm not so sure that it's a positive mouthfeel thing that you get. It's the diversity of the manoproteins on the surface that bring that kind of I'm losing the word for that, but it's it's like a pleiotropic effect. It's a lot of little things going on at the same time. It's not just we've never isolated one manoprotein that gives the mouthfeel effect, and I think it's the complexity of the yeast surface and the differences in all of those branched carbohydrates that are out there that bring that effect. So I'm not going to spend a lot of time on this. I don't want to get into a discussion on oxidation. I actually have an entire presentation on oxidation. So I'm just going to, a quick definition, chemical definition, oxidation, a hydroxyl becomes oxidized to a carbonyl, an alcohol to an aldehyde, and then it can be further oxidized to a carboxylic acid, just as an idea to see what that looks like with a specific molecule. Ethanol, you can see with its nice hydroxyl group here, can be oxidized to an aldehyde, where you've got your double bonded oxygen, and then further to an acetic acid, where you've got hydroxyl double bonded oxygen, COOH. And in wine, what accounts for a good deal of this is what's called the Fenton reaction, and it's iron dependent. There's trace amounts of iron in the fruit when you bring it in, and there's trace amounts of iron in your wine. And iron is a catalyst for the Fenton reaction, and it creates a peroxide radical. And this fellow is what goes around and transfers and removes electrons and does all the oxidative damage to molecules like your flavor aroma molecules that have specific little units like right here that when they're hydroxyls, they taste really good. And when they're aldehydes, maybe not so much. So with that in mind, that's kind of, sulfides, oxygen, catechol, quinones. Like I said, I don't want to belabor this, but I just wanted to show that there's an interplay between sulfites, oxygen, iron, quinones, catechols, just to show that it's a cyclic thing. It's not a one molecule to one molecule. And this has been, I want to mention these guys' names. This fellow, Danilowitz, who published the paper I pulled this particular data out of in 2011. He's published 2 papers very recently, and Andy Waterhouse has done some really good work recently to show the reactivity of these different components in the oxidative cycles in wine and the kinetics of their reaction. And to me, I'm real excited about this because if you ask winemakers, what do you really want to know? What's some of your biggest problems? Oxidation is in the top 3 every time, if not at the top. And there's all sorts of practices that go on in winemaking about trying to keep oxidation to a minimum. But what's really going on in the wine? Do we really know? And I'm on the American Vineyard Foundation review committees for Oenology research submissions. Basically, we fund basic research, and I'm on the review committee for that, along with a lot of really smart people. And 3 years ago, Andy Waterhouse submitted some work on doing some oxidation work, and first year didn't yield very much. The debate was, should we keep funding this? And the second year, we funded it, and it got a little more interesting. And the third year, he came out with all the kinetics on this stuff that I think is just going to make a huge impact in how winemakers make their wines, treat their wines, and limit the oxidation in wines going forward in the bottle. It's powerful information. And Danilowitz, with this work, outlining this particular grouping is also really important for that. So, real quick, redox potentials, redox tower. Here's people think SO2 provides a reasonable antioxidation effect. You saw in the cyclic area, it's a part of it. It's not really reactive. It's not going to react. The lower on this redox tower, the more reactive these things are in terms of redox reactions. So SO2, it's got a positive number. It's not bad, but ascorbic acid, more reactive. Vitamin E, more reactive. Tannins, 600 to 750. Tannins is a very diverse molecular set, so their number is not one number. But down here at the bottom is our friend glutathione. So just real quick, SO2, in wine, where does this stuff come from? We all know yeast can produce some SO2, and winemakers can add SO2. Okay? Tannins can come from grapes, they can come from oak, as well as winemaker additions. And glutathione comes from grapes, can come from yeast, and it can come from winemaker addition. Okay? So I'm going to talk a little bit about glutathione. I'm going to mention kind of what it is. Glutathione is it's the main cellular antioxidant in almost all living cells. I'll say eukaryotes for now. I'm not so sure about prokaryotes. I haven't studied it that extensively. But in all plant tissues, you have glutathione. It's a scavenger. It goes around and it scavenges up radicals that can damage DNA and cause mutation and create havoc in cells. Radicals can modify enzymes and limit their effectiveness. It's moving those electrons around in a haphazard way can create damage inside of cells, oxidative damage. This guy, this tripeptide, it's produced enzymatically. It's non-ribosomal, so two specific enzymes link these amino acids together. This particular one in a backwards linkage, this is glutamic acid, I believe, and it kind of caps this end. But you've got this sulfhydryl group from this cysteine that's in the middle of this tripeptide. This is the guy that does the scavenging right here. And this molecule, this tripeptide, there's an enzyme called glutathione reductase that's inside the cells, and it recycles glutathione. Glutathione goes around, it picks up electrons. Glutathione reductase relieves it of those so it can go back out and scavenge some more and protect the cells from oxidative damage. So as you can imagine, there's a little bit of this in the cells. Okay, it's not like one or two molecules. There's enough in there for the cells to protect themselves. It's kind of part of its immune system from internal damage. So if you try to add this directly to an aqueous solution or a finished wine, it gets oxidized very rapidly. And with no glutathione reductase to recycle it, it's of very little value. So this is one of the precursors for glutathione. It's the dipeptide. Basically, what we found with research on glutathione is by adding these precursors, cysteine, glutamylcysteine, and even whole glutathione, to the ferment about 2/3 of the way through. This is at the late stationary phase. Okay? If you add these things to the ferment early, try to give them to the yeast to get it to do this early, it'll break them down and use it for nitrogen. Okay? So you want it to finish its log phase and get into stationary phase and then get kind of late in stationary. Okay? And then when you add this, the cells are under some stress. Their metabolism is weakening. There's less substrate for them to grow on. Their energy is deficient. And they're under more challenges environmentally inside the cells. So they'll accumulate this glutathione, and it'll keep getting recycled from the glutathione reductase. And the effective concentration inside the cells can be driven up considerably. And just you may say, okay, well, there's lots of glutathione in all the grape cells and everything when it comes into the winery, and that's true. But if you've ever seen an HPLC profile of phenolics from juice, there's a huge peak. It's the biggest peak of everything in there, and it's this guy, GRP, known as grape reaction product. And what that is it's glutathione linked to caphtoric acid. There's lots of caphtoric acid from the plant tissue, the cell walls, and it gets released during crush and into the juice, and it reacts with the glutathione very rapidly. So that's where most of your glutathione goes that comes in from the vineyard. So just some quick numbers: glutathione in juice, and then glutathione in the corresponding wine. You can see that without any additions or supplementations, that the amount of glutathione increases from what comes in to what ends up in the wine. And that's because during autolysis, the Saccharomyces will put more into solution. So the glutathione that comes in the juice, it's proportional to your initial YAN. Your yeast available nitrogen, the amount of amino acid, nitrogen, and ammonia that comes in the fruit, the amount of glutathione is proportional. If you have a year with low YAN, you have low glutathione too, so you're at higher risk of oxidation in your juice initially. A lot of your, don't worry about that. I was going to talk about flavor aroma compounds in the juice, but that's a long conversation too. So, great glutathione rapidly lost through oxidative handling as well as the caphtoric acid binding. Good alcoholic fermentation nutrition, carbon-nitrogen balance, allows the yeast to release additional glutathione. The less stressed they are, the more they're going to do good things for you. And like it's the work on glutathione, I mean, we're just learning what these good things are. Besides the finished product and a nice flavor to your wine and yeast fermentation character that helps build the aromatic complexity that comes in from the vineyard, you've also got things like this that are contributing to the longevity of your wine and how long it lasts in the bottle and how fresh it tastes when you pour it. Glutathione and yeast can be supplemented. What we've seen in our experiments is that when this number is 20 ppm or higher, you get the protective phenomena in the bottle. I'm not presenting any product data here today, but we've got data that shows when this goes up over 20, that the freshness of the wine is increased farther out when you open it. Recent evidence. This was something that I did at a winery up in Napa Valley. We were using the glutathione supplementation, and this was in a biodynamic winery. And what they found is that when they supplemented with the glutathione, that they did their initial SO2, maybe it's not biodynamic because they were using SO2, but they had SO2, and what they found is that they could use less of it, that it stayed in the free form more. So back to that cyclic chart, you're challenging the SO2 less in the oxidative cycling of compounds in the wine when glutathione is there. Glutathione had the -9 or the +920 on the redox table. It reacts faster, so the SO2 is still there. The SO2 being 170 stayed longer than all of the others. So lesson in using less SO2 in your wines as well. So oxidation reduction, let's move to some selective adsorption. This is pretty straightforward stuff. It's just mass action. And basically what we wanted to show with this, with the kind of yeast hulls from autolysis and a couple of different proteins. Here's a globular protein and here's a more linear protein, gelatin. So what we wanted to show, this is reacting these preparations with Bradford protein reagent, basic brilliant blue dye. It's a charged molecule, and it interacts with proteins that have specific charges on their surface. You can see that the linear proteins didn't react as well as the globular or this matrix that is the cell wall. And then if you centrifuge these, low speed centrifugation, you'll see that the cell wall fragments will pellet, pulling the dye with it, indicating the protein is embedded in the cell walls, whereas these soluble proteins don't pellet, and you can still see the dye associated. There's more than one way up the mountain on all of these things.
When you said yeast would preferably jump on these before we would use any other nitrogen?
No. In the late stages, in late stationary phase, when the carbon is getting all burned up they will pretty much not be building any more. There's always turnover of proteins, and they're always using nitrogen. But there's internal cycling from the proteolysis as well. When you add these at the 2/3 of the way through, they'll take them up, and we haven't looked metabolically exactly how they do it, but when you treat the yeast this way, pull the yeast, hydrolyze them, and measure the glutathione, there's more there.
I was asking about grapes. If you increase it in juice, let's say.
Yes.
Is that going to be used right away by yeast as nutrient, or is it going to be preferred to use other type of nitrogen?
It would, like I mentioned, it would be nice to trial it and see if you could drive the level up, and then what happens to it. A good deal of it, you're going to see a GRP peak that's probably bigger proportionally to the amount of glutathione you bring in. And again, does it really stay? I guess if you had it at a certain amount, and you came with a nutritional profile that was solid, that your yeast were able to, because they're going to take up what they want to take up most easily. So there's, out of the 20 naturally occurring amino acids, there's groups of those that are preferred and some they don't like at all, like they don't like proline very much at all. It's hard to process. So I think whether the yeast were to use up that glutathione would depend on what their nutritional context was in the ferment. So the point with this particular slide was showing that there's protein in these yeast hulls and that it precipitates with the insoluble portion. And then basically taking a look at the properties of these, showing that with the differential zeta potential, that these things are fundamentally different. These, as being yeast hulls, the outer surface of cells, they're all pretty much negatively charged. But the interaction is different with these yeast hulls, and it's more of a hydrophobicity-driven interaction. It's different than traditional fining agents, so it does work a little bit different. And then some of the effects of this, the difference between a traditional, say, an albumin and yeast hull fining, you can see that the height of the lees is considerably less. The lees are more compact, which will be able, easier racking, less wine loss in lees. So again, real kind of basic mass action fining stuff. So that's 4 different topics related to Sur Lie aging and that process and defining more of what's going on and targeting specific molecules. And basically the way I look at this, since I started in the industry with Gallo in 2004, as a molecular scientist and not a winemaker or not an enologist, when I sat down with the winemaking teams that I've worked with then and also through Diageo and other teams, I sit down with the winemakers and I say, where's your pain? And then I think about what I know and say, can I help this? And the winemaking process I look at as, and the winemakers describe this as a black box. There's parts of it we don't understand. We do this and we look for these outcomes. And what I try to do, and I always have and I still do today, is I try to put handles on the outside of that black box. I don't ever try to shine light on all corners of it because winemakers like to have their art. But I can put handles on it and try to create more control points. And that way, winemakers can make stylistic decisions and have more control over the process as they move forward. And that's a lot of what this is about. It's trying to be able to provide more control and handles on the process. So, what else do we have to look forward to? Well, there's lots of information out there concerning lees and oak interaction. We haven't completely defined lees and malolactic fermentation influences, and the specific yeast cell wall membrane components and detoxification of must. Again, an important part of keeping everybody's ferment healthy through the very end. So there's lots of areas, I think, for more work to be done. For those of you that don't know, Laffort is a family-owned business based in Bordeaux, France. We've been around since 1895. It's the 5th generation now. Luc Laffort is the president of the company now. We're the #1 producer of enological products in the world. Based in Bordeaux, we're in over 50 countries. We're global. Sarco is our lab in Bordeaux. We also have a Sarco lab in Spain. Largest, best funded private research entity in the wine industry. There's other companies that serve the wine industry that have bigger R&D departments, but they serve bigger markets too, like biofuels fermentation, bread, brewing, lots of different businesses. We work very closely with the Bordeaux ISVV, the University of Bordeaux. We have faculty on staff there, and we fund students around the world, master's and PhD projects. And you can see the red dots are where we have sales offices. We're in China, and we're in Europe, and Australia, New Zealand, South Africa, and South America. That's me. This is Daryl and Natalie over here. So, I've been carrying this around with me for a long time, and I always like to put it up there. This quote says, "The task is not so much to see what no one has yet seen, but to think what nobody has yet thought about that which everybody sees." One of my favorite quotes. It drives me a lot in what I try to do research-wise. Anybody have an idea who said that? Who is this handsome fellow? No idea? That's our friend Erwin Schrödinger, 1933 Nobel Prize for Physics. So, with that, any questions, comments, discussion? Yes?
Can we just thank the speaker and then we'll go to questions? So, please join me in thanking Peter for a great Thank you very much. It's my pleasure to be here today. Questions?
So, on discussion on pre-ox and white Burgundy or Chardonnay wines, some people believe a lot of that has to do with whether it's whole cluster pressing or if it's batonnage. I don't know if there's any current information, but do you run into that at all in your research in batonnage and Sur Lie and pre-oxidation of wines?
So, Pre-ox to me it's a property of the wine that makes it, has a propensity to oxidize earlier than it should. It's not oxidized when you bottle it, but it's a property that means it deteriorates rapidly in the bottle where you'd expect it to last longer. Are we on the same page with that?
Yeah.
Okay. With that said, I don't know that we have any specific research programs looking at that right now. Unfortunately, I'm based in North America and our lab is in France. And you know, I don't know how many of you folks here know Laffort from 3 years ago when we were distributed through Scott Labs. And since then, Laffort has gone out on their own in North America. So we're a relatively young company. And that's one of my jobs, is to interface with universities and research people around North America and try to find areas of synergy where we can work together and investigate problems that are specific to our market or globally. And you know, with a young company, and I've got a lot of duties and technical support for all of the clients we currently have, and supporting our technical representatives. We have 4 now, based in California, and we have distributors around North America. It's been really difficult for me to be able to do that in the year and a half I've been with the company. Once a year, I go over to Bordeaux. We have what's called an export meeting, and I get a download there of everything that's going on. And you know, questions like this, bringing that sort of thing back to Bordeaux and creating an awareness there is part of what my role is. So no, I don't know anything in terms of defining that at this point.
Just to go back to the glutathione issue, you're talking about increasing yen and vineyard, and then looking at prospective numbers of the must harvest. You were saying, I thought I remembered a point, saying at the initial stage, or a must stage, that your GRP is relatively low. This is why, in addition to a must stage, you don't get the recycling effect. But then you made a comment that if you have high glutathione proportional to yen numbers, that you would have elevated GRP in the end. I'm just...
I think what he was saying at the start, Ryan, is that with typical levels of glutathione in must, it's a small peptide, depending on the concentration of nitrogen available in the must, the yeast can actually take that up and break it down and use it as a nitrogen source.
Right.
But also in the must, there are phenolic compounds that can bind up to the glutathione and make it not bioavailable to the yeast for any reason, whether it's as an antioxidant or as a nitrogen compound. But there's a correlation. The higher the must nitrogen, the higher the glutathione. But the question, the unknown at this stage, because this is a relatively new area of research, how much would you have to increase the glutathione in the grape, in the must, to allow it to have that protective antioxidant effect? They know it has an effect if they add it 3/4 of the way through the fermentation. Is there a way to boost it up in the must to still have that protective effect without having to add it later on? But I think that's an unknown question at this point from what you were saying. You just don't, the research hasn't been done yet to look at ways to increase the glutathione, through viticultural practices in the vineyard. And even if we did, up to what level would it have to increase, and how much GRP would have to be there to have the protective effect? Is that a fair?
Yeah, the GRP isn't going to, it's not a protective. It's kind of, I know, what would be interesting to know how much caphtoric acid is there as well, because you could make a correlation between the amount of caphtoric acid and how far you drive the glutathione coming in from the vineyard, because that's going to be a reaction that happens very rapidly. That glutathione is not going to be carried through to the end. The earlier you put it in, whether it's from crushing of fruit that's been enhanced in the vineyard for that effect, the longer you have to carry it, the lower your chances are you're going to pull it through. Like I say, the caphtoric acid is a strong scavenger at the beginning. Yeast, highly metabolically active, are a pretty strong user of all compounds they can get their hands on. Saccharomyces is not a notorious great nitrogen scavenger. It doesn't have proteases. So the things it can pick up, it grabs hold of pretty good. You could put all the protein in the world in a media. It couldn't use it for nitrogen because it can't break it down. But it can take up glutathione and its precursors. So it'll eat it fairly readily. And I would expect that with the way that ferments get pitched, the amount of yeast that get put in initially, it's a pretty big population. And the limitation is pretty much nitrogen from the beginning. And so they're going to soak that up a lot if it comes in from the vineyard. So like I say, I wouldn't put a lot of hope that that's going to drive your mgs per liter of glutathione over 20 in your finished wine. But like I said, it's worth giving a try. If you're a viticulturist and you're looking for ways to help, that could be a target, because it's fairly, you could take a small block or a couple of vines even and just follow it through the process and see what it does. The problem is measuring glutathione. Now, you can go online and you can find people that'll measure glutathione, and they'll gladly take your blood and do it. But they don't measure it in anything else. I went to the NFL, the National Food Lab, and they told me that it would be prohibitively expensive for me to want that done. In other words, they're not going to do it. And I got the same response from many labs. And we measure glutathione at Sarco Lab in France. So I work with people, and we set up trials, and we do things, and we capture samples and appropriately process them, and I send them to France for glutathione analysis. But I don't know here in Ontario what the labs do, and if there is an environmental lab that tests for glutathione that would test in wines or in musts. But it was tough in the U.S. to find one. I didn't find one. I tried to contract with a local environmental lab. I pulled the papers on the reactive compounds that bind with glutathione, and you could measure them. And the guy said he was building the assay, but it went on and on and on for months. I was like, hey, what are you doing? Oh, I'm almost there. It never happened. To this day, I never got result one. So the measurement can be difficult.
But does Sarco Labs, is it a public?
Yes.
So
Yes. Yeah, if you wanted to send samples there, they'd be more than happy to accept them. They'd be more than happy to tell you how to process them to preserve the molecules so that you're going to get reasonable results back.
And if nothing else, too, you're talking about that addition would make your sulfur additive crushing even just more effective for fermentation.
Yeah, it seems like during fermentation, the adding of this, and of course we're not, sulfur is minimal coming in on the front end, but after fermentation, after the first add of sulfur, in doing the cellaring, they found that they didn't have to add sulfur, that their free sulfur was staying where it was supposed to be. And so that was an observation. Now, we didn't ever say that was an effect of doing this, but when that particular winery told me about that, I said, well, let's set up a series of trials. And so we did. We set it all up, and then like 60% of the trials I set up in working wineries, it went to hell. It didn't work. The samples didn't get pulled. The wines got blended, and it's just what happens. 60% of the time when I set up trials, that's the way it goes.
Did you look at all in that research about the uptake of the glutathione in spontaneous fermentation versus pitched or inoculated?
No.
So you don't know if there's a difference in the rate of...
My initial assumption would be it would be slightly different, because what you're counting on is total yeast cells initially, and then the total mass that goes through autolysis and releases later. And I'm thinking, just I may be biased, but I get the feeling that native fermentations are not going to be as the cell populations won't be as high. They struggle a little bit more at the end of ferments. A lot of times natives aren't used to the level of alcohol. I'm thinking California here too, where we have some higher alcohols. If you're at 13%, I don't think there's very many Saccharomyces going to have a problem with that. I would just think that with a more homogeneous inoculum of the same yeast strain, which also I've done a lot of work on strain ID through ferments, that changes. A lot of times the guy you put in the beginning isn't the guy that finishes, or he's there at 20% at the end. But during late stationary, it might be 90%. But if you check at the end of the ferment, or even when you're down to one brix or so, or even less than that, the population is all bets are off as to who did the business during stationary phase.
One last question.
Sorry to backtrack a bit, but on the MP32 or MP40 kilogram, do you have any data on filtrability on wine after addition? Does it impair filtrability in the wine?
Yes. The data on the filtrability is, let me speak specifically to the MP40, what's known as our Manostab product. In the usage criteria for that, how the condition the wine should be in before you use it, because it's different. It's kind of a paradigm shift in stabilization. You don't substitute this for traditional cold stabilization after your ML. You've got your wine in your cellar, you cold stabilize it, and at some point in time you're going to go to bottle. When you use these non-subtractive technologies, you got to change your paradigm. This should be the last thing you do before you go to bottle. Okay. Now, if you meet all the criteria for addition, when you get to the point where you add it, put it in at the dosage, 48 hours, no filtrability issues. If you try to filter within 24 hours, you're going to see a decline in your filtrability. Wait 48, no problems.
Well, again, please, Please join me in thanking Dr. Solomon for his wonderful presentation.